All right, this is Bio 181, Chapter 15. It's a video on the central dogma of molecular genetics, which is really referring to creating proteins from DNA. Now, if you have any change in your original DNA, so say you've got a sequence, right? You've got a codon. Usually you're just going to copy that and not going to change, right? Every time we have this process of transcription, we're making an exact copy, right? But over time, there might be an error, and maybe one of the letters, one or more, changes. And so that change, gosh, where did that color go? There we go. Oh, just one color. All right, I'm not doing color. So that change is going to be a mutation, right? So anytime we have any variation in DNA, it's going to be a mutation. And that's the starting point of evolution. So the more variety you have, then you can have natural selection, which is going to select the individual that's most fit for the given environment. So I'm just going to go over several different types of mutations. A point mutation, that's what we just looked at. It was a single nucleotide change. So say you go from A and now it's replaced with T. A silent mutation has no effect. And in fact, most mutations in the genetic code are going to be silent. So we've got long sections in our genetic code that are not genes. They don't code for anything. They're just extra content. So like A, C, G, U, C. So if it's, and you've got a gene over here, here's gene 1, right? Then you've got another gene way down here. Okay. So if you've got a mutation here where it changes, say from G to C, no big deal, no effect. So that's a silent mutation. Even if it is in the gene, it often can have no effect. And this is one of the things, if you remember from learning about codons, there is a thing called redundancy. So if you have a sequence of, and that codes for a certain amino acid, right? You can actually have a few other triplets that will code for the same amino acid, which means it will have no effect. All right, missense mutation, you have the amino acid is changed. So say you have, you have a given codon and it's going to code for amino acid number 10, okay? And then now you have a mutation, so you have a new codon, and that new codon is going to code for amino acid, okay, number 20, okay? So it's going to look different. So maybe it was cytosine and now it's tryptophan, right? So maybe it was cytosine and now it's tryptophan. Okay, so that can have an effect. A nonsense mutation is where we've got a stop codon. So whereas originally, say you have this long gene and you've got it somewhere in the middle here, you've got a codon, say UCC, and then all of a sudden you have a mutation and it's going to code for one of the 3 stop codons. Then you have your, well, I won't draw the whole thing out, but you've got your, remember this is your DNA. Yeah, I'll move on to the messenger RNA. So this is your messenger RNA. Your ribosome's moving along, okay? And not getting my colors today, that's all right. So the ribosome's moving along. Once it hits that it's making an amino acid chain as it goes. So, but once it hits this point, it's just gonna disassemble the large subunit and the small subunit are gonna move apart, and then you have a half-baked protein. So that's a problem. Frame shift mutation. So this is where, you have codons. So A, C, C, you've got then C, C, U, U, G, G. Just making up 3 codons in a row. So this is going to be amino acid 1 in our sequence. This is going to be amino acid 2. And this is going to be amino acid 3. Okay. Now, if you have a change, some sort of a mutation, or say you add in an extra codon and it shifts everything down. So then now this is, say I'm going to add in a U. So now I have AUC as my first amino acid, right? And then this is shifted. So the next sequence that it's going to read is CCC, right? It's going to read these 3 together here, and then it's going to read these 3 together. So this is a big problem. So you're completely changing the amino acids that this is going to code for by shifting the reading frame down. And you could do it by adding in an extra 1 base pair or more, or you can delete base pairs. So these are pretty simple. I'll go through them quickly. So deletion, you delete a nucleotide. So it was AUG, right? So now it's AG. Okay. It's just missing 1. Duplication, you're adding in 1. So you've got AUG. So now it's A. Whoops, that's not an A. So maybe you have 2 A's, AAUG. Okay. So that's what we're talking about. Inversion, you've got a sequence reversed. So AUG, this actually happens quite a bit, is now GUA. Okay, so it's backwards. And then insertion, so we're adding in a nucleotide. So we've got AUG, and I just showed this on the previous one. Oh, insertion, it doesn't have to be the exact same though. So slightly different. So it could be A, U. No, that's the same. It could be A, C, U, G. Okay. All right. And it could be more than a single nucleotide. So you could have A, U, G, and then you could have a, let me see, in the middle here. So AUG is now A, and then I'm going to insert C, C, G, C, and then I have my UG, right? So here's what I had initially. Okay, and this is what's inserted. Oh, is that it? Okay, that's it. All right, cool.